Hello, everyone. We are going to begin 2.6, looking at the structure of DNA and also the structure of RNA. And this is a quick topic and kind of basic, but also very important. So here are the assessment statements. We have, sorry for a second, I'm getting used to this. So outline the structure of DNA in terms of a sugar, a base, and a phosphate. We're going to get a little bit more specific about what that looks like. So sugar, base, and phosphate. So the sugar is deoxyribose. No, there are 4 bases. So we'll go into detail on that. State the 4 names of those bases. So of those, we need to know that they're adenine, guanine, thymine, and cytosine. How they're linked together in a single strand, although we do know that DNA is made of 2 strands, but we're going to be talking about how they're linked together covalently in 1 strand. And then when we start talking about the 2 strands, where we get this famous double helix, which I think everybody's pretty familiar with the whole like windy thing. When we talk about 2 strands, how those are linked together with hydrogen bonds, one of the skills that you need to be able to do is draw and label a diagram of DNA, and they could be asking you to draw them in 1 strand, or they might ask you to draw them in a double strand. So make sure you read those questions carefully. And the last thing is we're going to compare the structure of DNA and RNA. So that is the plan. Oops, I made my screen too small. Okay. So let's begin with this. So I want you to remember that when we're talking about DNA, we're talking about the genetic material, and the genetic material you're very familiar with is probably a chromosome, right? And there's many different ways you can draw a chromosome. Sometimes you see them as like an X joined together with this thing in a centromere. Sometimes you see them all like squigglied up in this like supercoiled structure. So I think sometimes we lose the big picture. So when we're talking about this, we're talking about what makes up your chromosomes in your body. Now, if we looked closely at one area of this, we would notice that the structure of a chromosome is broken down in repeating units. Just like we were talking about carbohydrates being made of monosaccharides, just like we were talking about proteins made up of amino acids, DNA, our chromosomes are made of these things called nucleotides. So that is the 4th organic molecule that IB really wants you to know. So we have our carbohydrates, we have our proteins, we have our fats, and the last one is our genetic material in this thing called a nucleotide. So just real simple, a nucleotide is just made of 3 things, and they're listed right down here. So one of the 3 things it's made of is this thing called the nitrogenous base, which can be any one of those 4 bases. And a nitrogenous base, when we draw it, we draw it in this, I'm going to erase this real quick. We draw it in very simple like geometric shapes. So the nitrogenous base we draw as a rectangle. So there's my nitrogenous base. And the nitrogenous base is covalently bonded to a pentose sugar. So if you remember when we were talking about our sugars, they can be in the shape of a pentagon, not a very good pentagon, but okay. And we were talking about ribose, but in DNA, that's what the D stands for, the deoxyribose is our sugar. And then the last thing it's made of is a phosphate. and our phosphate is drawn as a circle. So we have 1, 2 covalent bonds here. One covalent bond joining our nitrogenous base to our sugar, to our deoxyribose, and another covalent bond connecting our deoxyribose to our phosphate here. Okay. Let's go to the next screen. I think I have to make this smaller in order for it to go. Okay. So here's another image of it. There's our circle for our phosphate, our pentose pentagon for our deoxyribose, our sugar, and also our base. And here's another more complex version of it. Two point 2.6.2 wants us to remember the names of our 4 bases. That's actually an HL requirement that they might ask you to list them. However, they constantly refer to them as adenine, guanine, thymine, and cytosine. So it's important whether you're taking SL or HL that we remember. But those are the 4. I do want to say one thing about these nitrogenous bases. So if we look at the structures of them, so if we look at adenine and guanine, they're listed as a group called purines. And if you look at the purines, which we have over here, adenine and guanine, you notice that they're made of double rings. So adenine and guanine are made of double rings. So one way that I remember that, if you're very good at your symbols on the periodic chart of the elements, Ag stands for gold, and you have a pure gold ring to remember that they're made of double rings. Maybe, if that helps. And then the pyrimidines, if we look at our pyrimidines or cytosine and thymine, these nitrogenous bases, they're represented by 1 ring. And remember, we always draw them when we're just doing a quick sample, or a quick illustration, we always draw them just as those rectangular shapes. But thymine and cytosine, they're made of 1 ring. They're our pyrimidines, and adenine and guanine are our purines.